Welcome student in the next lecture of our plant tissue system which we are discussing in our 4th week of the SWAM course Anatomy of Flowering Plants. In this we will try to discuss about vascular tissue system. Now vascular tissue system consists of the vascular bundles which are found distributed in the stele. So this all the other parts we have discussed that there will be an epidermis and then there will be a cortex. Okay. So then you will have the central part, okay, which will be surrounded by epidermis and then pericycle. In the central part, which is known as stele, you will see the vascular bundles. So vascular, see, when we are talking about stele, we are talking about the pericycle also, the pith also, the medullary rays along with the vascular bundle. But when we are talking about the vascular tissue system, we talk about the vascular conducting elements, that is the xylem and phloem, and along with that cambium, if present, it might be present or it might be absent. So xylem and phloem is the common tissue which is found in the vascular tissue. However, cambium may or may not be present. Now the function of this system is definitely conduction. Okay, we have already discussed that xylem is there which conduct water and minerals, whereas nutrients from the root to the different parts of the plant, whereas you have phloem which conducts the prepared food or the carbohydrates to even to the roots also, to the lateral branches, flowers and to every other part of the plant. Now the vascular bundles these are derived from procambial strands. So you have protoderm, and then you have procambium. So procambium will give rise to vascular bundles. So in vascular bundles you will have xylem, phloem and then cambium if present. Now how these vascular bundles are arranged in different plant types, that is also very important, and their distribution is quite variable. Like it is arranged in circular pattern in dicotyledonous stem. However, it is scattered in monocotyledonous stem. So when you will see a dicot stem, you will see that the vascular bundles are arranged in the form of a ring. Okay, so this is the vascular bundle, and when you will talk about the monocot stem, you can see there that it is scattered in the ground tissue, randomly distributed. Okay, so this is the vascular bundle and this is the ground tissue, and you can have in monocot, you can have hypodermis also. So now here you can see that the ground tissue is not differentiated into different layers also, like cortex and pith and medullary rays, endodermis, pericycle, but here you can see that the vascular bundle is lying in the central stele. However, some variations could definitely be there, so that all we will also study in anomalous structures chapter when we will discuss later in our coming modules. Like these vascular bundles could be also found in different other regions also, but it's a normal, but it's not a normal feature of a plant. It could be found in the cortical region. There, that time it will be called as cortical bundle, or it could be found in the medullary region, like in the pith region. So they will be known as medullary bundles. Okay, like in cortical bundle, example is Nyctanthus. In medullary bundles also, like in Boravia, in Bougainvillea, in Mirabilis, you will find the presence of extra bundles in the pith region or in the cortical region. Now, constituent of vascular bundle. See, the main important that we have discussed is the xylem and phloem and cambium. Now, talking about xylem, we have already discussed what is the structure of xylem and what are the components of xylem. You all know that xylem basically 4 components are there. You have tracheids, you have vessels, and then you have fibers, and then you have xylem parenchyma. So, all the structure and how they are arranged and functions also we have studied. Now, the first formed xylem is known as protoxylem. Okay. Now, when the plant is growing and when the plant is developing, the xylem which originates first in the growing tissue is known as protoxylem. Okay. Now, the composition of protoxylem is, it is made up of tracheid, vessels and parenchyma cells. Now, here you can see that fibers are not present in protoxylem. Why it is so? Because protoxylem is still growing, like it is still found in the growing tissue. So, it should have some flexibility that it can grow. So, fibers, as we all know, that they are highly lignified and having thick secondary wall. So, they do not have any possibility of growth. So, that time the fibers have not matured as such. And in tracheids and vessels also, see, that also we have discussed that tracheids and vessels also contain lot of lignification, lot of deposition of secondary cell wall, which make it hard and which actually make the cells die also. But when in the case of protoxylem, these tracheids and vessels will contain annular or spiral thickening. Now, this thickening, because they will help them in slight elongation. This thickening will actually help them to slightly grow. It will give them a chance to grow because many of the surface will not be lignified and this lignification will be found only in some small areas. Now, in case of protoxylem, the vessels and tracheids, like they will have smaller cavities, particularly like vessels, we talk about that they are very broad pipe-like structures having very broad cavities. So, in case of protoxylem, the cavities will be slightly narrower. Now, when the xylem will move towards the development of metaxylem, where the plant has grown little bit and it has slightly attained maturity, the protoxylem elements will be converted into metaxylem, means more now the formation of xylem which is taking place will be more having vessels having more bigger and wider cavities, and the vessels will also have reticulate and pitted formation. See, that also we have discussed that these are more complex formation which will actually make the cells more harder, and now they will not have any chance of elongation. So, now but definitely it will be formed when the cell have attained maturity and these cells are not dividing. It is not that every cell is not dividing, but in these elements these cells will not have a chance for further division. Now, coming to phloem. Now, phloem is in stem, it is found away from the center of the axis towards the periphery. So, that also that when xylem is present, suppose this is the vascular bundle, it is always that phloem will be on the outer side and xylem will be on the inner side. So, phloem is found away from the center towards the periphery. Now, this also we have discussed different parts of the phloem, that most of the components of a phloem are living. You have sieve tubes, then you have companion cells, then phloem parenchyma and phloem fibers. Okay. So, among them only fibers are dead, whereas other components are living. Now, even in phloem is known as protophlloem. Now, these contain narrow sieve tubes, fibers and parenchyma are found. But in this you can see that companion cells are not found in protophlloem. It is because when the phloem is still developing, it still have some of its organelles, some of its nucleus, but as it matures, the sieve tubes will lose their nucleus, and then a companion cell is required for its coordination. After protophlloem, when the plant matures, metaphloem develops. Now, the metaphloem is more complex and contains all the parts of a phloem. It also contains companion cell. Now, coming to cambium. Now, in xylem, a thin strip of meristematic tissue will be there, which is known as cambium. So, the cambium will also be developed from procambium. See, it's not the first form cambium. The cambium which will be first formed will be procambium, and then this procambium will actually give rise to xylem, it will give rise to phloem and also the cambium. So, this cambium is a very thin strip, generally a single layer of tissue which is found in between xylem and phloem. So, if this is the xylem and the outer one phloem, this is the xylem, and then you will have cambium. Okay. So, it has meristematic activity. Now, phloem and xylem are not meristematic, but this cambium will have the meristematic activity and will perform the function of division and will give rise to secondary tissue. It is found in dicotyledonous stem. So, that is why I have told you that cambium might be present or it might be absent. So, dicotyledonous stem will have cambium, because in dicot you would have observed that these stems have a very big diameter, they are very strong, and it is because of this cambium that it will divide and it will give rise to secondary tissues which will increase the diameter of the plant, and that is why cambium is also known as lateral meristem. Now, the cambium strips will be generally uniseriate. See, generally it is single layer, but sometimes a multi-layered cambium could also be observed. These cambium cells are living, okay, because they have the function of division, and they will be elongated and almost flat tangentially, means they will be elongated, okay, but they will be flat on this surface. Now, coming to different types of vascular bundles, different arrangement of vascular bundle which is found in plant parts. Now, this is a picture which shows, see, this is a vascular bundle, okay. Here also you can see, although it is a microscopic image, it will be slightly different from the image which you generally see in books. But these are also depicting the same type of tissues that I will tell you later what are they. Coming to the first type, that how these vascular tissues are arranged. Now, when we say that it is the radial arrangement of a vascular bundle, see, it is found in dicot and monocot root. Now, when we call it radial, that means vascular bundles are present in patches, in different patches of xylem and phloem. So, xylem and phloem patch. So, here you can see that they are radially side by side, okay. Xylem is forming a different patch and phloem is forming a different patch, and this arrangement is radial. That means xylem and phloem both have different radii, okay. And when we talk about conjoint, so in this case This is found in dicot and monocot stem. See, when we take one single vascular bundle, we will observe that xylem and phloem are found on single radii. So, this is the phloem and this is the xylem. So, this type of arrangement is known as conjoint. Okay. Now, in conjoint also and closed. Now, open vascular bundle, we say it when cambium is present. Okay. This is the phloem, this is the xylem and this is the cambium. Now, this cambium is this type of situation, this is known as open vascular bundle and it is found in dicot stem. Now, we say it open because in the presence of cambium, secondary growth formation will occur. So, you can remember in that way that it is open for secondary growth formation and we call it as closed if cambium is absent. Okay. I will draw a very thin line just to demarcate the presence of phloem and xylem. So, phloem is there and xylem is there. Okay. So, no cambium. So, this type is found in monocot stem because you would have seen monocot stem that they are very thin, they generally don't have very thick stem because of this absence of cambium. See, those stems also in many cases they are not so large also, like wheat and rice or maize. You all might have observed the stem of these plants and they have a very thin stem. Then you have collateral vascular bundle. In conjoint also we can say that they can be collateral, means when phloem lie together on the same radius in which phloem is outward and xylem is inward. See, this situation is definitely radius, it is lying on same radius, but the situation could be opposite also, like if we can label this as xylem or this as phloem, but when xylem is on, phloem is on the outer side. Okay, in this situation, okay, it's better to understand here because this is the outer side. So, phloem is on the outer side and xylem is on the inner side. So, that condition is known as collateral. Okay, and then you have bicollateral. See, they are also open bundle, means definitely cambium will be there, but in this case cambium will be present on both the sides. Okay. So, in this case you can see here that two cambium is present on the outer side between phloem and xylem. Okay. Suppose this is the xylem and this is the phloem. On the inner side also between xylem and phloem you will see two cambia. Now, this type is found in the members of cucurbitaceae. Now, another type is also there, concentric. Now, coming to the next type, that is, now concentric vascular bundle means in which one type of tissues completely surrounds the other type of tissue. Okay, like either xylem is completely surrounded by the phloem or phloem is completely surrounded by the xylem. Now, in this also, like you are having concentric circles. So, in this case also you have two forms, amphivessel and amphicribural. In amphivessel, in this xylem surrounds the phloem. Now, suppose this is the phloem and this is the xylem. Okay. So, this is not a normal occurrence of vascular bundle. So, definitely it is not found in every common type of plant parts. It is found in some plants like Dracena. So, that we will discuss later also this the composition and the organization of Dracena stem, Yucca stem also. Dracena is basically a monocot and it has anomalous structure because monocot does not have a secondary vascular tissues, but in Dracena secondary vascular tissue occurs and these bundles occur in this form that xylem completely surrounds phloem. So, this is known as amphivessel. And then you have amphicarybril in which phloem completely surrounds the xylem. So, this is phloem and this is xylem. This is also not a very common type which is found in higher plants. It is found in some ferns, okay, some pteridophyte it is found. Now coming to another important organization of vascular bundle on the presence of protoxylem or on the basis of arrangement of these xylem elements. So, xylem, or as we know that it is we can divide them into protoxylem and metaxylem. Okay, that is the first form xylem, okay, and the later form xylem. But when these xylem tissues are getting formed, it's not that they will be completely lost. See, they will be formed when the plant is growing, when the plant is increasing in size. So, these elements will also be there, but they will be now occupied by more bigger structures. Like protoxylem will be also there. When you will see the section of a plant under microscope, you can see that protoxylem is there, but it will be now added by metaxylem. Metaxylem, a broader space will be occupied by metaxylem and both the tissues will be present together in a xylem. So, now how these two different types of tissues are present in xylem, that is very important, and on that basis different arrangement has been proposed for these two tissues. Now, different arrangements are there like endarch, exarch, these are most common type. Now, when we say it, suppose this is suppose the central cylinder, central steel is there, okay, and in this case, suppose we talk about root, okay, in root we have sorry. This is an exarch arrangement and this is an endarch arrangement, okay. Now, exarch arrangement we call it when protoxylem is on the outer side and metaxylem is on the inner side. That means protoxylem is towards periphery and metaxylem towards center. Okay. So in this case, you will see this type of arrangement which is known as exarch. See, from the name itself, you can slightly try to remember it that see, on the basis of protoxylem we are talking about an exarch or end arch means when the protoxylem is on the inner side. So end means you can just, for the sake of your just remembrance, towards the end of the this tissue organization will be the protoxylem and outer will be the metaxylem. So you can remember that when protoxylem is towards the end or in the center and metaxylem is on the outer side, it is end arch condition, and when protoxylem is on the outer side and metaxylem on the inner side, it is known as exarch. Okay. And exarch condition you will find in roots, whereas end arch situation you will find in stem. However, the organization will not be like this. In stem you will have, see, phloem is there, then cambium is there, then you will have xylem arrangement like this. This is the stem. So this is the stele. Okay. So this will be the proto and this will be the metaxylem. Now, you will have another condition which is known as mesarch, in which protoxylem is surrounded by metaxylem, but mesarch stems have multiple. In mesarch condition, you will have protoxylem in the center. In this, see, in mesarch condition, you will have protoxylem in the center and surrounded by metaxylem on the outer side. Okay. So here you will have protoxylem and the size of the xylem will increase on both the sides. So this will be metaxylem. In mesarch condition, you will have a various vascular bundle, and centrarch is also similar to that of mesarch, but you will have only one vascular bundle, whereas in mesarch condition you can have several vascular bundles. So that is the difference between them. Now a small assignment I just want to do it here. Just see, this is a structure of a, see, basically it's a dicot stem, but detail we will study later. But that also we all have discussed about all the different tissue types. I just want you all to label them and just try to identify those tissues, like what is this one and this part and this part, and also try to differentiate them into tissue system. Try to identify the tissue and try to identify the tissue system. So for this I will end this lecture here. Thank you.